Today we're going to talk a little bit about DNA replication. DNA replication is when DNA is copied, and this only happens before a cell divides. So this whole entire process that we're talking about today only happens every once in a while in a cell when it divides. Here's what you need to remember about DNA before we talk about how it divides. You need to remember that DNA is a really long strand, and that strand is made up of something called a nucleotide. And that nucleotide is made up with a phosphate, a sugar, and a base. So that nucleotide portion has a phosphate, sugar, and base. And those bases are A and T and G and C. And A and T's are always going to match up with each other, and G's and C's are always going to match up with each other. If that's a little bit confusing, you might want to check out the tape on DNA, and that could help you a little bit. You also need to remember this. DNA is wound up really, really tight in an organized fashion called the chromosome. If you're still a little bit iffy on chromosomes, you might want to watch that tape, and that could help refresh your memory also. Alright, so we have DNA, and it's wound up into chromosomes, and now we need to have that DNA replicate so that as a cell divides, it can now cut in half. So we're basically doubling DNA to cut it in half, which makes sense because then we'll end up with the right amount of DNA, I should say. So here's where it all starts. This whole process of DNA replication starts way up here at this guy. This little box area is called an origin. Origin, the word, means the beginning. And just like origin, the word means beginning, origin, the place in DNA replication, is the beginning of replication. It's where this entire process starts. We are going to look at just one origin right now, but in actuality, in one chromosome, there are tons and tons and tons of different places where we have origins, where we have replication happening. And the reason why is because if we have more places where replication is happening, it can happen more quickly, and the cell can divide faster, which would be more efficient for the cell. So we have an origin. That's the place where something called helicase attaches. This guy right here is helicase. Helicase has this funny ending, ASE, at the end. Whenever we see a word in science that ends in ASE, we know right away that it's an enzyme. And an enzyme is just a fancy way for saying a protein. So helicase is a protein or an enzyme that unzips the bonds between the bases. So this is how I think of it. Say we have two strands of DNA, right? And those two strands of DNA are stuck together through these bonds. Just like a zipper is stuck together through the teeth of a zipper, right? And so if we have something like a zipper that comes and unzips that strand of DNA, that's the same way that a zipper will unzip the strand of a zipper that you zip up on your jacket. So a helicase would be like a zipper. Alright, so we have helicase that's going to come along in a strand of DNA. So here's our strand of DNA. And helicase is going to come along and it's going to take these bases apart from each other. So now they split. So now we have one old strand of DNA and we have one new strand that we need to make of DNA. Because remember, DNA has two strands. So now we have the one side of DNA. So now we need to form the other side of the DNA. And we do that through an enzyme again called polymerase. Polymerase ends in ASE, so you know right away that it's going to be an enzyme or a type of protein. So polymerase is a special enzyme that's going to attach those nucleotides or bases in, just floating around here in the nucleus. It's going to attach them like kind of like a stapler, forming one old strand of DNA and one new strand of DNA. So this guy's going to move this way, this guy's going to move this way, and soon we're going to have two completed new strands of DNA. If you look at it, it's kind of making a Y shape, right? And so this whole area where this is occurring is called the replication fork. It's like a fork in the road where you have one path and it suddenly splits into two paths and you call it a fork in the road. It's the same exact thing, except this is a replication fork where we have different enzymes interacting to make new strands of DNA. Now there's a couple other things you should know about this. This whole process is called semi-conservative. What that means is one strand of DNA is going to be old and one strand of DNA is going to be new. So we always have an old strand and a new strand of DNA. In our example over here, this was our old strand of DNA, and this is the new strand of DNA that's forming as a polymerase attaches those nucleotides. As DNA is replicating, 50 bases are added per second. Think about that. That's crazy. We have 50 of these guys that are attaching every single second in just that origin. And now we have tons and tons and tons of origins that are happening on a chromosome. And we have tons and tons of chromosomes that are happening in every single organism, right? And so just think about how many bases are being added all the time. Because this is happening so quickly, there's going to be some mistakes, right? Maybe a T doesn't match up with an A, or maybe a G doesn't match up with a C as it should be. So what happens is, once in every billion nucleotides, an error occurs. So in every billion of these, a mistake might happen. Repair enzymes are constantly checking to make sure that every single nucleotide that is added is not that mistake, because mistakes can be detrimental. If there is a mistake and it isn't caught, it's called a mutation. We a lot of times think of mutations as being bad, but in actuality, all mutations aren't bad. In fact, mutations are part of the reason why we have natural selection today. They can be detrimental, though, including some genetic diseases, which we'll talk about more another time. So overall, DNA replication is where DNA makes a new strand of DNA from an old strand. He starts at a place called an origin. We have an enzyme called a helicase that unzips the old strand of DNA. So one strand is old, and then a polymerase will come along and add that new strand of DNA. It's called semi-conservative because we have an old strand and we have a new strand. 50 bases are added per second, and every once in a while we'll have a mistake that happens that we call a mutation. So hopefully that helps, and if you have any questions, as always, let me know.